21. Lessons learned. What has the saga of the discovery of anti-cancer drugs taught us? The reigning image for all diseases in Western culture is that of war. It is in the area of cancer that military metaphors are most often applied. We speak of how the disease strikes or attacks, setting in motion the body's defenses. We speak of struggle and resistance in the battle against cancer. Modern medicine hunts for magic bullets in the crusade or war against this malignant foe. In exploring the cultural mythologizing of disease in her book Illness as Metaphor, writer Susan Sontag condemns the militaristic language around cancer as simultaneously marginalizing the sick and holding them responsible for their condition. Yet one overwhelming fact persists. Cancer will kill more Americans in the next 14 months than perished in all the wars the nation has ever fought, combined. Biomilitarism, however, oversimplifies the problem by clouding the fact that cancer is a catch-all term for a class of more than 110 separate malignancies. One military metaphor labeling America's cancer campaign as a medical Vietnam proved to be woefully apt. Francis Moore's congressional testimony in 1971 and the Institute of Medicine's analysis on the war against cancer were sadly prophetic. No clinically important scientific breakthrough, Moore argued, had ever resulted from such directed funding. It is clear that programmatic research dictates unhealthy rigidity that inhibits creative minds and constrains a critical feature of the discovery process, serendipity. The more narrowly a scientific goal is defined, the more the discovery process is obstructed. Scientific discoveries so often happen when they are least expected. Cancer has for too long been treated with some combination of surgery, radiation, and chemotherapy in a program derided by some as slash, burn, poison. In this war on cancer, in which all cancers were considered the same, this was a scorched earth policy. A new understanding of this enormously complicated disease was desperately needed. Fortunately, by the 1970s, molecular biologists made revolutionary insights into the characteristics within the cell that lead to the initiation of cancer. But these resulted from determined efforts in the fields of cell biochemistry, tumor virology, and medical genetics. There are more than 10,000 billion cells in the human body. In most tissues, cells are continually dying and being replaced, and control of the equilibrium is critical in maintaining the normal architecture. The complex biological blueprints for the behavior of cells are controlled by their genes. Humans have 20,000 to 25,000 distinct individual genes, and genetic information is carried in DNA molecules. The human genome is estimated to include 3.08 billion units of DNA. The complexity of what the cell's machinery can generate is indicated by the fact that a few thousand genes can make trillions of combinations of proteins. Cells multiply 10 million billion times during the course of each human lifetime. Mutations, random changes in the design of a gene's DNA structure, occur constantly because there is an intrinsic error rate when DNA is copied. In extreme errors, the cell will self-destruct. Otherwise, the aberrant cell survives. Starting in the last decades of the 20th century, sophisticated genetics and molecular biology have been aimed toward a more precise understanding of the cell's mechanisms. Yet even here, chance has continued to be a big factor. Surprising discoveries led to uncovering cancer-inducing genes, oncogenes, and tumor-suppressing genes, both of which are normal cellular genes that, when mutated, can induce a biological effect that predisposes the cell to cancer development. A search for blood substitutes led to anti-angiogenesis drugs. Veterinary medicine led to oncogenes and vaccine preparations to tumor suppressor genes. In one of the greatest serendipitous discoveries of modern medicine, stem cells were stumbled upon during research on radiation effects on the blood. Experience has clearly shown that major cancer drugs have been discovered by independent, thoughtful, and self-motivated researchers. The cancer war's guerrillas, to use the reigning metaphor, from unexpected sources, from chemical warfare, nitrogen mustard, nutritional research, methotrexate, medicinal folklore, the vinca alkaloids, bacteriologic research, cisplatin, biochemistry research, sex hormones, blood storage research, angiogenic inhibitors, clinical observations, COX-2 inhibitors, and embryology, thalidomide.